Hello, I'm Professor Claire Steeves. I'm a professor in Ageing and Health, and I'm the clinical director of TwinsUK at King's College London. Thanks very much for joining us.
Hi there, my name is Dr. Ruth Bowyer. I'm an early career researcher, and my research focuses broadly on how environmental factors influence ageing and health in ageing. And that's what we're here to talk about today, isn't it, Claire?
Yeah, that's right. And you've been involved with a lot of the work that we've done on ageing over the years, Ruth. So we're delighted to introduce this webinar with the motive of celebrating the 30 years of amazing collaboration that we've had with all of you. It's a true milestone, and it couldn't have been possible without all your support in doing questionnaires and in coming up for visits to help us really study ageing and health. And if you've followed all of this webinars that we published this week, you'll know that the number of scientific and health questions that we've contributed to have been really amazing. And that's all down to your efforts. It's been really impressive. On this webinar, we're going to be focusing on all of the valuable research that you've been doing and supporting with ageing. And we want to answer some of the questions that you've sent us, which show your interest in this field. I mean, it's not surprising really, because the population is aging fast, and being able to age well and preserve our health is, of course, very important to all of us. So that's really what we've been all about in sort of TwinsUK ageing research, is about trying to understand how we can keep as fit as we can. Before we start, Ruth, how exactly do we measure ageing in TwinsUK?
Yeah, it's a great question, because on the face of it, it's an obvious question. We all know what it is to age, right? But actually, there are very different ways you can measure aging, and that's because aging can happen differently in different parts of the body. So to understand how someone is aging, we have various different types of measures we can take. So they can be biological age measures, clinical measures, or kind of an interim between the two as a measure from imaging. And that's really why it's so valuable that we have all of you volunteers coming into the clinic so we can get these measures, because it's something that you can't ascertain from questionnaire data alone.
Yeah, we're really excited that in the next year, we're going to have a unique opportunity to understand how the structure of the body is really aging. And we'll be able to look at that in comparison to all of the very detailed measures that we've taken over the last few years that map biological aging. So we were talking about a few of those just now, Ruth. Let's talk about some of the ways that we sort of map biological age within TwinsUK.
So we can estimate your biological age looking at various different markers, and these markers we can often ascertain from information we get from your very valuable DNA measures, so the DNA that we get from you when you come into the clinic. So they can be things about how your DNA might be altered due to factors that relate to exposures you've experienced across your life. And a really great potential example of this is how we see some lifestyle factors interacting with aging differently in different organs. So, for example, we know that obesity accelerates aging in the liver specifically, but we don't see that in other organs. And perhaps a different example is how sunlight is a really strong factor which contributes towards the aging of your skin, but it doesn't necessarily mean, for example, your cardiovascular health would decrease. And actually, you might see the opposite, because potentially the reason you have UV damage to your skin is because you're going outside to exercise a lot.
Yeah, really interesting. And so those markers on the DNA, I mean, some of them that we've studied, and we've studied right since the beginning that they've been investigated and understood, is things like telomeres, the caps on the end of chromosomes that in a sense protect the chromosomes from more wear and tear. And as divisions go on, we know that these telomeres, they tend to shorten, and we can measure that in white blood cells, and that provides one marker. But there's another marker which I know you've been working at, but also Jordana Bell, who's in the department. She's one of our very top researchers, who's one of the best in the world looking at epigenetic age. So these are markers on the DNA which we've identified and others have identified are really strongly associated with age, and also with conditions that occur in aging, so like cardiovascular conditions and so on. And so using these clocks, these epigenetic clocks, we can look at how people have aged over time. And then we can compare them to how people feel, how people experience disease, and also things like the structural aging in their body that we'll be hopefully getting with the MRI study that we'll be doing from next year.
So Claire, we've spoken a little bit about how obesity only accelerates aging of the liver, but not other organs. But are there any other examples that might be interesting to learn about in terms of how aging might differentially affect other organs in the body?
Yeah, so that's really interesting. Obesity and aging in the liver, that's relating to epigenetic age, that we see that epigenetic age signature ticking away in the liver differently in relation to obesity. But of course, obesity does affect other systems, not just epigenetic age. So that sort of underlies the importance of looking at multiple different markers of aging at the same time. But also, as you rightly point out, we have to look at multiple different different body parts, because actually it was the twin study that helped us show back quite a long time ago now, your twins study, your twins data, which showed that different body systems don't age at the same rate. So, for example, if we look at how the eyes age, they don't age because of the same underlying issues. They don't have necessarily even shared genetic factors that contribute to the aging within them, and say, for example, your bones and how they age, or how your cardiovascular system ages, with, for example, blood pressure changes with age. And so we have to look at all of these different systems separately. And that's where coming up to a visit is really important, because we, in the visits, as you know, if you've been to one, we look at your blood pressure and your cardiovascular responses. We look at your muscle strength and your muscle ability to walk and to rise from a chair. We look at your eyes in quite a lot of detail, because we've got a really great aging research group that's led by Professor Chris Hammond. And we've got measures of your lung function, which changes with age. And then also now we're doing the imaging study. So we're going to be looking at lots of different aging across lots of different body systems within that. And that allows us to look at aging in multiple ways and see how each of us as individuals are aging differently over time. And the idea there might be that if we could predict that using biomarkers, we could tell people what specific actions to take that are relevant for their own aging pathway. And I think that's really important from the point of view of clinically what we're going to do going forwards, is if I know that my problem is osteoporosis and muscles, then the action that I'm going to take to try and avert that future aging is going to be different from if I thought my problem was going to be relating to the liver, for example. So does that answer your question, Ruth?
It does. Thank you.
So you've been looking at how diseases sort of club together or affect people at the same time, haven't you, Ruth? Do you want to say a little bit about that?
Yeah, that's right. So it's a really kind of key area for us to study, because some people, as they age, they can remain very healthy, and live a very long, fulfilled, healthy life, whereas other people acquire a series of multiple long-term conditions. So that can be when you have multiple different diseases at the same time, and that can really decrease people's quality of life, not only because they suffer from the conditions themselves, but also because it means they have to take a lot of different drugs in order to treat those conditions. And then because they're taking lots of different drugs for lots of different conditions, these drugs can interact with each other and cause that individual more health problems. And we also see in other individuals that perhaps but as they age, they're either more or less robust to sort of challenges from their health. So if you think maybe of an older person in your life who's had a fall, perhaps, sometimes that fall can really lead to longer term consequences. So they don't bounce back and recover from that. Whereas other individuals might have a fall, they might have to take it easy the next day, but really there isn't a longer term consequence that's easier to see. And that's really what we want to help address in twins, isn't it, Claire? We want to help address the health span rather than the lifespan.
Yeah, that's absolutely right. And twins, your work has been really crucial to demonstrating that these biological aging measures mean something in relation to clinical outcomes. And so we've been in all of the papers that you look at, for example, in epigenetic age, we're there supplying data. Your data that you've given is there providing that evidence. But one other thing that we can obviously help with is understanding how this changes over time and how this develops going forward. And that's something that's less well studied. And it's having a longitudinal data set, which is quite rare within the grander scheme of things, that has these measures over time. That's where we can really add a very specific benefit in terms of understanding aging. And then if we can understand aging, we can start to look at mechanisms that might underpin it. And that's where your work comes in around environmental work, Ruth. So should we just talk about that kind of debate about genes and environment and aging?
Yeah, it sounds good. So I guess there's probably an idea often that there is either your genes that result in a health outcome or result in whether you get a disease or not, or it's potentially an environmental factor. But you might have heard the nature versus nurture debate. That's often what it's called. But really, we all kind of, I would guess, agree that it's nature via nurture. So your genes interact with your environment to lead to a disease outcome. But obviously, your environment varies a lot, throughout your life and depending on who you are. And that's one of the amazing reasons that we can use twins to answer these questions, because twins share a lot of the genetics, either all or half, depending on if you're identical or non-identical twin, but they might have different environmental exposures. So to give you an example of the different environmental exposures that can influence how you age, some recent research has sort of summarized a lot of the existing knowledge that's out there and has found that green space within the area that you live in, low exposure to pollutants from traffic and air pollution are associated with longer telomere lengths. So those telomeres that Claire was talking about earlier, which are these little things on the end of chromosomes, and we can measure to associate with your biological age, those things are longer and therefore suggestive of a better biological aging in areas of low pollution and higher green space, so higher exposure to natural environments.
Yeah. So it's really good that you've been able to give both the genetic information from being a twin pair, but also genetic information from those samples, but also a range of other measures as well as questionnaire data and information that is related to, for example, where you live, which will help us kind of really tease out these environment and genetic contributions. And of course, those biological age measures, some of them we think are better at tapping the sort of environmentally driven aging. And so, for example, we look at metabolites in the blood or the degree of sugars sticking onto your proteins that are really good markers of aging as well. And we can look at them all together with those other markers that maybe are more trapping genetic changes over time. So all of this work, I guess, it's all about trying to address things in the future, help people to live more healthy lives going forwards. And this is where twins can be really, really useful, actually, because in some situations, it's possible to do a real randomized controlled trial and actually test whether or not something works to change aging. And I'm going to tell you a little bit about how we used one of those within twins and used the specific benefit of twins to really help to do that easily over the pandemic. But some other things, like, for example, you were talking about pollution and so on, Ruth, or maybe some other environmental exposures. It's not really very easy to design a randomized controlled trial on that. So what kind of ways can we use twins and your amazing twin data to really understand these really important environmental effects? Ruth, do you want to say a bit about that?
Yeah, definitely. And yeah, just to add to what Claire was saying, it's such an important time to be considering how we age and how the environment affects us, because we are facing an increasingly aging population. There's going to be many more people over the age of 65 in the decades to come than has proportionally ever been before. And coupled with that, we're facing extreme climatic breakdown, and that affects all parts of the environment. So whether it be the ecosystems we inhabit and the associated biological entities that inhabit those ecosystems, or whether it just be as simple as the exposures that we face, such as a rising heat or higher levels of pollution differently affecting us. So it's so important to be considering this, but as Claire says, pretty impossible to design a kind of using a real experimental design, because what you'd have to do is you'd have to separate a load of individuals and then, in one room perhaps pump in a load of air pollution, and then the other room pump in some lovely clean air, which would not be very ethical and would not be in any way sustainable long term. And so using the magic of twins, we can use a quasi-experimental design. So that means using individuals who share genetics, but perhaps different environmental exposures. So for example, in some of the work we've done, we've looked at twins who live in different areas of deprivation. So one area which might have low environmental deprivation, which means there might be lots of access to green space, there might be low crime rates, there might be lots of access to education and employment, versus their co-twin who might live in an area of higher deprivation, so the opposite of all of those things. And because we know those individuals share the genetics, we can pick apart where it's the environment that might be resulting in a difference between the health outcome. Yeah, but we also have some really amazing real experimental designs, don't we, Claire? Where we're using a sort of more traditional approach.
Yeah, that's right. But before I go on to that, I was just thinking about all the work you've been doing as twins within the COVID pandemic, because you've been telling us about your experiences over COVID, and we know that COVID pandemic has been very different for different people. It's not been the same storm for every boat, as it were. Some people have been affected very severely by COVID itself, or maybe by the changes that went in hand with it. So some people, really got fit and went outdoors and took advantage of that. Some people weren't able to do that because they were heavily shielding. Some people changed their diet in a positive direction, others the other way. So we're going to be looking at all of that valuable information to really understand what's happened in relation to that and in relation to future aging. And the reason being, of course, is so that then we can understand what things that we need to do in general in order to make sure that we age the best we can. But onto that intervention study, Ruth, that you were kindly sort of prompting me to talk about. So over the pandemic, some of you took part in an amazing study, really, which was a really groundbreaker, which was led by my clinical doctoral fellow, Mary Nee Loughlin, who's an incredible young physician who's a geriatrician and wants to sort of really understand how we can age better using trials. And so she did a randomized controlled trial with you. So some of you came up, and rather you didn't come up because it was in the COVID pandemic. That was one of the reasons why it was so amazing, that some people in the trial were willing to take a probiotic medication together with protein supplements, and other people took a placebo and protein supplements, and they didn't know which of those two groups they were going to be allocated to. So the idea of this trial was to see whether or not adding a probiotic to protein can help improve an older adult's response to that protein. And the reason why we really wanted to do that was because we know from lots of other studies that older people don't really respond to protein that well. They don't increase their muscle mass or their fitness or their ability to get up and down the stairs as well as younger people do. And we think that might be because their gut microbiome is different, and that the microbes might actually be changing the way that the body absorbs or interacts with the protein. And so that's what Mary set up. And she's done a trial within the twins. And because we've had twins that have both done different arms of the trial. We've got a very sensitive design because, of course, those twins, they share the same age, they share a lot of their genetics, and they share a lot of early life environmental factors that contribute to aging. So using twin pairs within that kind of design, we can use quite small numbers to really detect effects, both on their physical health and on their mental health. So look out for the results of that are coming out soon, we're really excited about. So these are some examples of how, I guess, your work has really contributed to understanding more about what happens in aging and what might be the solutions that we could go for going forward. And there are definitely things that we can do as individuals. For example, physical fitness was something that I showed within my PhD was really important for cognitive aging. And many of you were heavily involved in that study. But also dietary changes and the gut microbiome and its relationship with how we age, making sure that that biological garden is as fit as it can be, so that we don't have translocation of bad bacteria into the body, which then can stimulate the inflammation that might lead to aging. And but the third thing is being able to do these investigations, these sort of interventions where we change something and we measure whether or not it's changed the health in the twins. That's a really valuable thing to have contributed to. So thank you, and thank you for your future engagement.
Yes, thank you so much. And just if you're interested in hearing more about the diet and the microbiota, there is going to be another webinar on that topic available soon.
Very good. Well, it's been lovely to speak to you, and I hope you've enjoyed the webinar. Do keep your questions coming.
Bye.